So I'm going to be demonstrating how to do cross-matching before blood transfusion. Today I'm going to be using the Rapid Vet H major cross-matching kit. I have my donor sample right here, and then I have my recipient sample here. The first step I'm going to do is take this recipient sample and put it in the centrifuge, and then I'll be back for the next steps. So now I have my recipient sample that is centrifuged. I'm going to take the donor sample, and I'm going to put 2 drops of that into the blue blood prep tube. I'm putting 2 drops of the donor sample into the blue blood prep tube. And I have that one into this blue tube now. And now I'm going to pick up some of the recipient's plasma and put that into the yellow reaction tube. I'm going to put 4 drops of that into this tube. One, 2, 3, 4. So 4 drops of the plasma is now in the yellow reaction tube from the recipient. Now I'm going to take 2 drops from the blood prep tube, and I'm going to put 2 drops in each of the other tubes that we have. So I'm going to start off with the reaction tube. So using the blue prep tube, 2 drops. 1, 2. Next, I'm going to put 2 drops into the negative control tube. One, 2. And now I'm going to put 2 drops into the positive control tube. One, 2. So I just finished adding 2 drops from the blood prep tube to each of these other tubes. Next step is going to be just gently inverting them to mix them. Now I'm going to let these sit for 5 minutes and then come back and move on to the next step. So these have been sitting for 5 minutes now, so I can move on to the next step. So the next thing I'm going to do is going to be taking 1 drop and putting them to their matching color. So I'm going to take 1 drop from the yellow reaction tube and place it into the yellow reaction tube gel. Take 1 drop. And just placing it into the gel. Now I'm going to do the same thing with the negative control tube, taking 1 drop from this tube and putting it into the negative control tube gel. Each time I'm doing this, I'm using a different pipette. One drop into there. And then the same thing for my last one, which is the positive control. Taking one drop from the positive control tube and then placing that into the positive control gel. My next step is going to be to take the tubes containing the gel. I'm going to centrifuge those, and now I just finished centrifuging the gel tubes. And to move on to the next step, I have to make sure that the negative and the positive control tubes are giving me the right results. So right here I have my green negative control, and I'm looking to see if the cells are on the bottom for this one. And I can see that the red blood cells are at the bottom of this tube, so that means that that one is correct. This next one I have my positive control, and these results should be kind of more towards the middle of the gel. And looking at this sample, I can see the red blood cells are in the middle of the gel area. So now that both of my controls look good, I can go on to looking at the yellow reaction tube gel. And for this one, this will tell me if the results are positive or negative, so if the patient is able to have this transfusion or not. So looking at this one, I can see that the red blood cells are towards the bottom of this tube, which is meaning that that is a negative reaction, so that this patient is okay to continue with the blood transfusion from this certain donor. So since this test came back negative, that means that we can go ahead and do the transfusion. If the test came back positive, we would see that the blood was located more around the matrix of the gel, and that would mean that the patient would be more at risk from that transfusion. So we would not go through with that transfusion from that certain donor to recipient.